Hi, and welcome to this lecture on environmental adaptation of animals. On this second video on nutrition, feeding and digestion, we are going now on the feeding. So we now know that, okay, what kind of nutritions we have, and wouldn't it be good that if we have some kind of information that, okay, we need now more proteins or no more fat? Well, we all know that it's not working on humans because we have these TV dinners, so we prefer sugar and fat. But it's not the rule for all the animals. For example, mountain gorillas, they are attracted to eat rotten wood to get more sodium that they need. And these can be tested. The balance of nutrition can be tested in wolf spiders that are very simple animals. And they are not making a net, they are hunting the prey by running. And they eat small insects. And if they are fed with fruit flies, they easily get either too much lipid or too much proteins. And these spiders tend to balance their diet by eating the flies with the nutrients lacking in their food. So if they yesterday were to having protein-rich flies, now they are eating more lipids. If they were having yesterday more lipid-rich flies, now they are eating more proteins. So these spiders are wiser than men. Okay, what kind of food we can absorb and how we can get it in our intestine? This is a killer whale. This is, you all know, and it's a very typical predator. Okay, it's not running after the prey, but it's swimming after the prey. But even in the same kind of environment, we can have several different feeding styles. So this killer whale, they are eating a little bit smaller prey than the whale itself. Then we have blue whale. That is a suspension feeder, so it's ingesting organisms that are very minimal in the size comparing their own size. And then we can have animals like reef-building corals that are photosynthetic because they get the nutrition from the symbiotic algae. And this feeding style affects drastically on the mouth structure. If you ever have been looking on the skulls of different animals, you have seen that, okay, they have all the vertebrates, they have different all the mammals, they have different kind of teeth, and in the birds they have different kind of beaks. And the teeth can be either used to cut the food in smaller parts, like in mammals, or grasp the prey. So the fish, even the predator fish, they have a very sharp but quite tiny teeth. So they are not used for cutting, but to grasping the prey and swallowing it. And the birds are very well known in the variation of the bills. Okay, they can be used, these bills, to get the prey from the tree trunks, or crush the sheets, or collect the prey in the deep mud. And why we have this kind of variation among vertebrates? Well, because the vertebrates have neural crest. It's one of these embryonic cell layers. It only exists in the vertebrates, and it's quite flexible. So it's affecting on the facial structures and on peripheral neurons. And it has been tested in the birds that if you take the neural crest from normal duck to put it in squill, you will get the peak of the duck, a bill of the duck. So it's affecting directly on the bill structure. And because it's also easy to modulate, then it's affecting that, okay, the birds that were migrating in Galapagos Islands, they were getting different kind of beaks. But also in the invertebrates, there is a variation in the feeding apparatus. And on these, the snails is a very good example. The basic structure of this feeding apparatus is very simple. They have a Radular apparatus with a teeth that is having chitin to rasp some organic material with their digestion. And it goes back and forth. So it can the apparatus can go down to grasp some material from this substance. And so it goes up and down all the time and to get some particles. But the structure can be got a little bit harder with using some iron or some silica. And it can be getting so hard that they can be also carnivore to drill the shell of the clam. So it's they can use the same kind of ribbon, but now not only eating some algae, but now they can eat other animals. And one of the examples of this variation is that it can be also done to produce this kind of harpoon. So there's a cone snail that is having this tooth to chop the fish and insert some toxins to immobilize the animal. And so we can have very large variation also among the invertebrates. And if they have the toxins, well, usually these toxins are affecting on the membrane proteins and usually on the nervous system. And usually only for a certain time, because the predator don't want to be killed himself. And we must remember that, okay, not only the predators are using the toxins, but also the toxins can be used for defense. So we all know that, okay, the plants are producing secondary metabolites to prevent eating. But some animals can detoxify it or tolerate the chemicals or excrete them. So it always with these chemicals, it's always some kind of fight or war going on. That, okay, will someone be tolerating this toxin? But then we have these suspension feeders. This is a blue whale. It's about 30 meters long, and it's eating 2 or 3 centimeter long krill. So they must eat more than one prey at a time. But actually, they can be quite, it's actually very good strategy for them. Because then the suspension feeding, then this food chain is much shorter. So from the same amount of algal cells, you can get much more food. Because even in effective food chain, about 10% of the nutrition can be used to growth of the small fish, and then for the medium-sized fish, then for the large fish. So that's why they can get a lot of more nutritions for the whale if they are eating small prey. And for this same reason, not only the large whales, but also the largest fish, like whale shark, are using the same strategy. And also about 1/3 of all the biomass of commercially caught fish are suspension feeders. And because this suspension is a little bit odd to us humans, we must look it a little bit deeper. In the whales, there are 2 sets of balance plates hanging from the upper jaw, and the inner jaw is frayed in the fibers. And the huge mass of water is taken in the mouth cavity and forged on each side of balance arrays. And if there's something else than water, if there's krill or copepods or small fish, they cannot fit through the mass of these 1 meter long fibers, and then they are swallowed. Okay, that's how it works on very large animals. What about then on fish? Okay, you can see that, okay, here is the gills, and the gill filaments are used for gas exchange, and they are combined in gill arc. And inside these, they might have some racons to concentrate the particles that they are swallowing. And this means that, okay, when they are getting mouthful of water, the water volume is decreasing because it's flowing through these gills. And then the food particles are more concentrated when they are going from the mouth to throat. Then the third way to eat or get some food is to use symbiosis. And we have different kind of ways to use the symbiosis and the microbes. We can use heterotrophs, so some microbes that are not producing the energy by themselves. And then we can use autotrophs that are collecting some other forms of energy for their these biochemicals. So we can have a photosynthesis or chemosynthesis. And on these photosynthesis algae, they are quite common in aquatic animals. And they use always, or actually practically always, some other feeding mechanisms. But actually they don't need it for the energy, because these algae can provide them as much sugar as they want. And very common example of this is reef-building corals that have in the gastrodermis, there is these algal cells that are getting sunlight and then producing sugars. But then we also can have these chemoautotrophs, like this annelid worm that is about 150 centimeters long, and it has no gastrointestinal tract or anus, so it can't eat at all. And it lives in symbiosis with a chemoautotrophic bacteria in the bottom of ocean, where there are some volcanic activity. And there's a cold seawater containing sulfur, and when it's going deeper in the ground, then this sulfate is reduced to sulfur. And then the sulfur, heated water with the sulfur, is going out from the soil. And inside this animal, the gills pick up the sulfur and some oxygen and deliver these with their circulation. And they have an organ called trophosome, where there exist bacteria that are obtaining energy from oxidizing this sulfur to sulfate. And this gives everything that this worm needs. But still, okay, these are quite exotic animal examples, but when we are thinking on our gastrointestinal tract, we have different kind of ways to do it. So some animals can have a batch reactor where they swallow some food and process it, take the nutrients, and then get rid of all the waste products. And they must get rid of the waste product before getting the next meal. And then we have this continuous flow reaction like humans. So first we have breakfast, then we have lunch, then we have dinner. And they can be all at the same time in our reaction, because they are getting inside from on one side and then going out from the other. And then we have the third way. So we can have this continuous flow reaction that takes a little bit more time, and the old material and the new material are already always mixed. And this is something that happens in normal composting, but it also happens in ruminants. So we can have heterotrophic microbes that are very common in our intestine. So more than 1 century ago, it was known that some animals have gut structures used for microbe-based digestion. And they are in an anaerobic environment, and they carry out fermentation. And we can have these foregut fermenters, like in cow or African buffalo. So we have these ruminants that have the rumen, where it's full of bacteria and some plant material that this bacteria is cutting in smaller pieces. But also this kind of strategy is used by kangaroos and some monkeys and even some birds. And how they work? Okay, from the esophagus we can get on the stomach, and in the beginning of stomach in these animals, it's non-acidic. It's used for the fermentation. Because if it would be very acid, then these bacteria would not grow there. And so in these foregut fermenters, they are fermenting first, and then later on there is this acidified portion of the stomach. So the good points are quite obvious on this kind of thing. First of all, we can't digest all the food particles. So we could use some symbiotic microbes to do it. And a classical example of this is, for example, cellulose that's cleaved by microbial enzyme cellulase. And then these microbes can cut also some plant structural carbonates to modulate them in short chain fatty acids. So we can expand our diet on more on plant material that it would be impossible to digest without these bacteria. Then these microbes also synthesize B vitamins, and it's also essential for so producing also some essential amino acids. So then we don't have to worry so much on what kind of nutrients do we have them more, because it's if we have this foregut gut symbiosis. And moreover, we have already thought that if we get too much proteins, we will get nitrogen waste, and that's sad, because in several ecosystems there is a priest of nitrogen, and these microbes can actually recycle this nitrogen, because otherwise they would be excreted by kidneys. But beside these foreguts, we also had hindguts and midgut fermenters. So there can be some enlarged cecum or colon that is used for fermentation, like in rabbit or pony. So it can be a long cecum and a dead end part, or then it can be enlarged column for this fermentation. And even then you can use the cellulose, but the problem is actually that the beta vitamins, the B vitamins and the amino acids, for those transportation you require some transport proteins. So probably in these cases the microbes are not able to give you any of these. And that's affecting that these hindgut fermenters may eat the feces. And then we have also midgut fermenters in some fish. Thank you.